basically what I'm doing here, okay. So basically what I'm doing here today is trying to give you an understanding of my research endeavor. Basically, we'll just be analyzing the research finding. Then we'll be buttressing and going through each of the methods and the methodology that was used. So you can see the possibility, and it's not just about not for knowledge sharing sake, but also so we can also check and also give suggestions if there are better ways of doing this and all of that. But we'll be learning in the course of that. I mean, we'll be having two practical sessions. I think the first is graph pad analysis and also image J quantification. So graph pad is a a software that we use to analyze, just like we have SPSS, R, and so on and so forth. So that was also what was used for this study, and we'll be making use of that. All right. Then for the image J is a software, a kind of a graphical image editing software, which we use for scientific purpose. So photographers use it too, but in our own end, it was used for the quantification and also some using immunoratio and all of that and cell counting. So we'll be doing that too, and during the course of the meeting. All right. So please just give me a minute so I can share my screen. Okay. All right. Can we all see my screen, please? If you can see, you can just indicate, please.
We can see it.
All right, yeah. Thank you very much. So I'll be starting just right away. Like I said earlier, my name is Tolulope Gbayisomore, and the whole concept is all about Phyllanthus Amarus, the role in mitigating potassium dichromate-induced locomotion, posture, and coordination impairment in male Wistar rats. All right. So this was actually my one of the projects that I was involved in at the laboratory. I forgot to mention, I think she has mentioned that I'm research assistant at the laboratory for experimental and translational neurobiology, which work with rodents, animals, C. elegans, that's Cenevitis elegans, and basically we do immunochemistry, neurobehavioral tests, and all of that. So this particular project was done during my BSc, and we were able to do some part of the laboratory studies in the lab when it was still at the Federal University of Technology, Akure. So, and these are my collaborators for the work. All right. So let's just do a quick one on the abstract. We got the rats. I'm just trying to explain in simple terms now. We got a rat. We induced with, after the pilot study, we induced the potassium dichromate. We assess the effect of the neurotoxicant, which is potassium dichromate. Potassium dichromate is more like is a chromium. Yeah, we know chromium and all of that. They are melters that evilent chemicals that are very detrimental to human health. Yeah. So potassium dichromate was used because we saw that a lot of studies have been done using chromium, but we wanted to see the effect, and also considering that chromium is more prevalent in these parts of the world where we have a lot of industrial engagement and all of that. So the main role was chromium, and when we looked at the molecular size and all of that, we saw that chromium really was a very pivotal part of the neurotoxicants. So we checked the cerebellar function, then we administered that Phyllanthus Amarus liver extract, then we assessed the function, and we did a couple of tests to check the results: the neurobehavioral tests, neurotransmitter assessments, antioxidant assessments, and immunohistochemistry. Basically, that's just a summary of it. All right. So what we'll be doing today, basically, we'll be taking the introduction. That's for us to have a background understanding of why this study. I mean, what is this study all about? Then the problem statement. Why are we doing this study? What is the problem we are trying to address? These are key aspects that set up the framework of we conducting any research. All right. Then the aims and objectives of the study, what we are focusing on, then the materials and methods, that's the methodology, then the results, the conclusion, acknowledgements, and references thereabout. All right. So introduction now. This whole study was focused on the cerebellum, and I'm very sure most of us, I think we are high school students. I don't want to go too complex, but I believe that we all know that cerebellum is one of the very, very important part of the brain that is responsible for motor movement, motor coordination, motor function, and a lot of locomotion activities. So this whole study was focused on motor conditions like Parkinson's disease, autism, thereabout. So motor conditions like the motor neuron disease pose a very, very significant risk on we living as human beings, with about 4.5 per 100,000 people being affected. And the very bad aspect of it is that there's an increasing trend over time. So let's also look at ataxia. Ataxia is prevalent in children with about 26 over 100,000, and a prevalence rate of dominant hereditary cerebellar ataxia of 2.7 out of 100,000 people. That means in about 100,000 people, you're going to see 26 people that's ataxia in children, and you see 2.7 for hereditary cerebellar ataxia, that's 2.7. And there's an increasing prevalence rate. So, and one thing about it is that this is a burden. Parkinson's disease as it stands, I think we all here, we are Africans. And the truth is that Parkinson's disease, these diseases they are understudied compared to the most common diseases that we see around, like tuberculosis, malaria, and thereabouts. So these diseases, most of them are understudied. And the truth is that when we have the problem, we are expected to solve the problem in the African way. Yeah, there are a lot of things that we ourselves can incorporate, and let's see what can work, basically. So out of the modern chemical drugs that of natural plant product origin, and especially in drug development of the pharmaceutical industry, there is need for more research endeavors, especially focusing on the orthodox medicine in treating neurodegenerative disorders. So this is the plant Phyllanthus Amarus. I don't know if okay, let me on my video. I don't know if we are quite familiar with this plant, but it's a plant that is very common in this part of Nigeria. Yeah, it's very, very common. And it's a tropical plant. We call it gale of the wind, stonebreaker, seed under leaf, and they have, it has Yoruba name, it has Hausa name, Igbo name, thereabouts, and so on. So we're just trying to see what are the mitigating effect on cerebellar impairment. And based on studies that have shown, they've reported high antioxidants, that's flavonoids, terpenes, and so on and so forth. Those were the common antioxidants that we're seeing in the plants. Then that's memory enhancing, anticholinesterase, estrogen, and also and the phytochemical analysis of the plant extract revealed contains a high amount of saponins, tannins, flavonoids, and alkaloids. All right. So now, what is the statement? Why are we doing this study? The neuroprotective nature of Phyllanthus Amarus against oxidative stress has been identified, and so the specific dose range for this neuroprotective effect against neurotoxic agents is poorly understood. That's one thing regarding most of the studies who do not make it to the clinical trials, like knowing which dose, okay, this dose they are talking about, how is this going to correct this issue? Yeah, we've seen that it can correct it, but at what specific dose? So that's one of the questions. And the only way to address that is when we do a lot of exploration and also repeatability of our studies. Like that is, if I do the study here, you in that site or in another region, you're expected to do that study. That's what's called science. The moment I conduct the study and you're not able to repeat it, although considering other conditions, then it's no more science. So science is all about we repeating the study. Then right there, we can now proceed. Okay, let's test these clinical trials and so on and so forth. All right. So the specific dose for mitigating these neurotoxic agents is poorly understood. And understanding its effects on potassium dichromate-induced cerebellar damage is yet to be documented. So we saw this knowledge gap and we felt that, yeah, this is something we really need to study. And so far, so good. The aim of the study was aimed at providing novel information about the neuroprotective effect of Phyllanthus Amarus in potassium dichromate-induced neurotoxicity in the cerebellum of male Wistar rats. All right. So the objectives, will be going through all of this with time. We assess neurobehavioral activity using open field tests, T-maze, spontaneous test, Morris water maze. Then we also conducted assess the level of oxidative stress. That's oxidation. Oxidation. I believe we have knowledge about what oxidation and anti-oxidation is all about. All right. So these markers, that's glutathione peroxidase, glutathione reductase, they are antioxidants, which we can use to know, okay, if there's oxidative stress in the brain, or the antioxidative level of or antioxidant enzymes in the brain are at the higher end. All right. So then we also conducted neurotransmitter assessments. There's GABAergic and glutaminergic pathways assessing that area. Then immunohistochemical visualization, neon and TNF-alpha expression. Then, so the animal grouping. So I think this is why we are here. The methodology aspect is why we are here, and it's going to take a major part of our time. Yeah. So for the animal grouping, we had 7 groups. We had the control, which we administered normal saline. Normal saline is from the word saline. So we purchased that from a pharmaceutical shop. Yeah, a pharmaceutical shop. And we got that. I think we used about 4 or 5 of that during the preparation. So we got that. Then we administered about 0.5 ml to each of the rats orally, daily. Then the study lasted for 20 days. I should have mentioned that it lasted for 20 days. Then the Phyllanthus Amarus only group, which we administered 300 milligram per kg per day orally. Then the potassium dichromate 70 milligram per kg orally. Then donepezil. Then we had high Phyllanthus Amarus and the low Phyllanthus Amarus. Now, what this did was that in most of the research, it's advisable that when you are trying to study maybe a neuroprotective effect or ameliorative effect, it's more advisable to have it at 2 different doses, the high, if possible the middle, then the low. So that way you are able to compare the effect, because the moment you just have one group and you are comparing it against maybe your control, that's you administered water or normal saline, then you might not really be able to see the comparative mitigative effect, right? So for the high Phyllanthus, we administered 400 milligram per kg. Then for the low Phyllanthus Amarus, we administered 200 milligram per kg. Then donepezil at 5 milligram per kg. Then the potassium dichromate ultra was 70 milligram per kg. So the question is, how did we come to this? So the 70 milligram per kg was based on understanding that the last or the closest study that we could see regarding potassium dichromate was actually a study where they administered it to fishes. Yeah, to fishes. I remember vividly. And it was a kind of so we had to conduct pilot study, and we also had to compare the leather dose before we could come to that dose, because like we all understand, there are a lot of, when it comes to using animals, there are ethical concerns regarding it. So we need to ensure that we do not pose the animals to a lot of damage, so that they are not they don't just die. Yeah. So that's one of the major things that we should also take to note. All right. So at the end of the day, we ended at 70 milligram per kg. Initially, it was supposed to be 50 milligram per kg, but at some point, we saw that if we wanted to get the expected effect, yeah, after the pilot study, we just had to leave it at 70 milligram per kg. All right. So for the methodology, I think we've described this before. We collected the Phyllanthus Amarus plant samples, moving to the aqueous extraction, 28 days administration, and behavioral assessments. Then animal sacrificing. Then we conducted immunohistochemistry, biochemical assay, GPX and glutathione reductase. Then it's neurotransmitter assessment of glutamate and GABA. So each of these is what we'll be analyzing in the forthcoming slides, each one of them. All right. So the first part is the aqueous extraction of Phyllanthus Amarus leaves. All right. So the leaves I showed you the other time, just like every other leaf that you have outside there, we collected it from a swampy area. Yeah, in an estate actually. So they have a farm there, and we got there and we're able to get as much as we can. We're given authorization by the guards there, and we immediately took it for identification, right? By plant biologists at the Department of Plant Biology at DME College of Education. And they gave it a batch number and they confirmed that, yes, this is the plant we want to use for our research. Then after that, after it was identified, we need to go for authentication. Yeah, so the difference between identification and authentication is that a plant biologist will help you identify your plants so that you yourself you can know. Because we took about two different plants. Then they told us that this is Phyllanthus Amarus, then this is Phyllanthus Noruri. Yeah. So due to the fact that we were focusing on Phyllanthus Amarus, there was a Phyllanthus Noruri, but the Noruri was what we wanted to use was Phyllanthus Amarus. And moreover, Phyllanthus Amarus is more common in this region than the Phyllanthus Noruri. Though they are of the same family, but different kind of species. So after identifying it, we took that Phyllanthus Amarus to the herbarium curator, and plant authentication was done, and it was assigned a herbarium voucher label, and we preserve the voucher specimen for future reference. So in a scenario where you, for example, now you want to get the plant that we use for this study, it can be easy with this number to go and get the plant and give it to you so you can replicate what you need to replicate. So the harvested fresh leaves were then shade-dried for 7 days with regular sun drying for better grinding. So we sun dried it, and we ensure that the sun is not directly sitting on the plant. So that's one thing every plant biologist or people in the area of plants will always tell you, so that the nutrients or what make the plant that plant, based on what you've read in the study, are not gone. So the dry leaves were then grounded into a coarse powder using a grinding machine and kept in airtight conditions for extraction. So the type of grinding machine that you use too is also important, but I won't go deep into that for today. So after grinding, we kept in airtight conditions for extraction. So now the extraction, we got about 600 grams of the dried material measured, and one thing is that you need to ensure that you get as much plant as possible. Yeah. So if you can get this much plant, you grind it, and what you're just getting is just small. That happened to us. So we had to go back to get more, then grind again. So after you grind, then we got it was macerated in 6 liters of distilled water for 48 hours at 4 degrees Celsius in a refrigerator. So the bottle contents were sealed, kept at room temperature. And the filtrate was kept in a stainless steel at 42 degrees Celsius until it dried up. So the extracts, the resulting extract after those process, it reduced from 600 grams to 13 grams. So we now place it in a small glass tin and stored at 28 degrees Celsius in an incubator for further studies. So the different doses of 200, 300, and 400 were reconstituted. We had to separate it, what we're going to use for, because we're using it for 28 days. We needed to ensure that it was divided and stored in a store bottle for later administration via routes to experimental animals. So one thing I would like to mention is that actually this 30 gram was not enough for the animals. I think we got to 20 thereabouts. Then we saw that it was finishing. So we had to quickly go back, divide ourselves into team, then go back, get more, then take the process again. So I think this study in particular allowed me to understand that proper preparation is very, very much needed, and also getting alternatives on ground. So assuming we had alternatives, we would have come short. But when we noticed at day 20, we knew that by day 25, this thing would have gone, and it might affect us. So the plan for 28, imagine we're stopping at 25, then it's no more to what we expected. So at that point, we had to go. Then before day 24, we had it, then we continue. All right. So the chemicals and reagents, normal saline from Biomedical Limited Nigeria was purchased from Uche Care Pharmaceuticals. Then the camel potassium dichromate 500 gram, molecular weight 294.18, and the UN number 3288 was manufactured by Danjin Camel Chemical Rangers Limited, and we got it through Pyrex Scientific Company, Benin, Ondo State. So then donepezil was manufactured by Mutual Pharmaceutical Philadelphia and purchased from Nutricare. All right. So the experimental animals, the experiment was conducted at the Department of Anatomy fully in terms of, because there was animal house, and even the whole process of the extraction was conducted there. We only visited the herbarium for the identification and the authentication. So the animal handling, animal kill, because there was an animal house there. Yeah, that's the animal house breeding colony of Unimed. It's also located at the Department of Anatomy. So we filled the animals with blended livestock in Ibadan, Nigeria. So choosing the kind of feed to give them to is also important. There's some feed you give to the rats, one way or the other, it affects their normal physiological structure. And sometimes, especially when you're looking at generational study, it affects their genes, one way or the other. You might not really know, but it's one way or the other, there are some mutations occurring. So knowing the kind of livestock feed to give them that fits them too. So approval was granted by the Research Ethics Committee of the University of Medical Sciences of the state with this approval number. Then the animals received human care following the principle of human care and the use of laboratory animals, and the rats were weighed weekly before the commencement throughout the experiment using a weighing scale manufactured by Sunrise Technology and calibrated in grams. So following identification, 70 young adult male rats weighing 100 grams and 120 grams were divided into 7 groups of 10 animals each. So we did this randomly, just ensuring that we had equal number of representation. So ordinarily, we made use of 80. Actually, we had 80 adult Wistar rats. So we had to keep 10 in case anything happens. We learned our lesson during the extraction. So we didn't want, during the preparation, so we didn't want, so we had 80 rats, and but being reported here, we made use of 70 rats. All right. So this is okay. So for the motor function test, so the whole idea was that when we started administration, we were supposed to conduct neurobehavioral tests. So there are specific neurobehavioral tests that we do, and considering that we are looking at motor coordination, we are looking at locomotion, which are the roles of cerebellar. There are specific established neurobehavioral tests that you can use. Yeah, and basically, you're expected to purchase them. I think there are companies that have produced them, but you yourself, you can create yours. That's with the help of people that are specialized furniture makers. They can help you design an equipment based on the standard protocols. So if you want to design yours, you can check it on YouTube. I think Maze have the dimensions. You can make of that. So for this, we use wire hanging test, open feed test, and the team is spontaneous alternation test. So I'll take it one by one now. For the wire hanging test, what we did was we started at day 3, and basically wire hanging test is to assess the muscle strength of the rats following the drug exposure, be it the neurotoxicants, the control, that's the normal saline, or the plant itself. So it's basically to assess the muscle strength of the animals, and also one way we do this is assessing the suspension time. So as suspension time is the duration by which the rats release itself from the wire. So that period is being recorded, and it helps to identify neuromuscular impairment or abnormalities. So the whole study goes this way. Before you conduct the study, before you conduct neurobehavioral study, it is recommended that you try to acclimatize the rats, because you are bringing them from an animal house, which they are used to this environment. Come on, just like me, for example, now. Let's assume I'm a specimen, or that you want to conduct an experiment on me. I'm used to this environment, right? I've been eating here and everything. Then suddenly you want to conduct this neurobehavioral test, and you are taking it out there, and immediately I start the test. One way or the other, I won't show you who I am inside there. That's just how it works. So it's recommended that you acclimatize a night before, right? And also, specifically for the wire hanging test, there are procedures, but we had to adhere with this particular procedure. So we trained each of the rats on the second day of the experiment so that they can get themselves familiarized with this test. So we started on day 3, or on day 2. We not only acclimatized, but we trained them so that they can get themselves used to at least have an understanding of, hey, bro, this is what you are expecting. So, and after that, the test was conducted day 7, day 14, and day 28. Then, and the cutoff time was set at 180 seconds. So those that got that were able to withhold the wire for 180 seconds, where that's the maximum basically. So if a rat dropped from the wire within this time frame, the duration by which it dropped will be noted. For those who completed 180 seconds, you just mark it. Okay, 180 seconds. Correct. All right. So the next is the open feed test, and this is how it looks like. Yeah. So this in particular, we have the rats in the open feed test. Then this is also the open feed test, but we have figure 1, a open feed equipment, then figure 1, the number of objects recognition test conducted on open feed test platform. So the whole idea there is that when constructing, you can use the same equipment for open feed for novel objects, right? So what you just do is ensure that after you've done that, you create a kind of a line that you can remove, right? So that line is used for the open feed, and for the novel object is basically used to assess memory, cognition, memory and cognition. So you have 2 sessions. I'm not going to go deep into that now, but you have 2 sessions where you have two different objects of maybe different shape or different color, one way or the other, but there has to be a difference. So the first stage, both same color, same shape. Then maybe the second stage, different color or different shape, during the case. So for open feed test now, this is how it looks like, and it's basically 100 by 100 by 100 by 100. That should be kind of equal. Then some people also make it of 75 by 75 by 75. As the case may be, just check the standard protocols. So this was what we had. Then we had a dark room. The dark room, as you can see, is in black and W. Then we had a camera on top. So, and everywhere was closed. So the moment we start, it starts recording. So that was how the open feed test was done. So what does this open feed test do? The open feed test is used to measure locomotion and anxiety. So a square wooden arena, 72 by 72 by 20. Some use 100 by 100 by 30, as the case may be. But the major thing is, once you divide it, you have 18 by 18 centimeters square. So the open feed test apparatus was cleaned with alcohol. So after you conduct your study, per each rat, you must ensure you clean the surface, because one thing is that when you put the rats in that open feed, most especially, the rats get to urinate, and urinate or fecabuli, those are also measurements on their own. So you have to clean it up, because another rodent coming in might just smell this and and it's going to be on that place all through. So it's not going to be able to move. So you need to ensure that you clean it with cotton wool and glycerol, or 70% alcohol. I mean 70% alcohol, not glycerol. Sorry. That's cotton wool and 70% alcohol. That is, the alcohol must not be too concentrated too, because you need it to dry. I mean, you don't want to spend all day there. And also, one thing you need to know for open feed test is that during the open feed test, we ensure that we start early, 6 o'clock, 7 o'clock. Once it's 11, 12 in the afternoon, we stop. So the whole idea is that in the morning, the rats they are eager, they are okay. I mean, in the morning, everyone is up and grateful. We are on our feet, man. But in the afternoon, they are tired. One way or the other, they are most times stressed up. So especially when we are brought them out, I mean, they've acclimatized themselves to the environment. Then now you've started, but this group, maybe they're just hanging there. So maybe theirs is starting around 2 o'clock, for example. at that point, the result you expect to get, you might not get it. I mean, the activity that this particular group who did theirs at maybe 9 o'clock in the morning, and this group who is just coming to do theirs around 2, 3, you might not get the same results. So these are things that we take into consideration. So we ensure that latest by 11, 12, we pause or we are done. So anyone who we are not unable to complete in the morning, we wait till probably in the evening. So they get in the evening, or we wait the next day early in the morning. So those are the things that we took into consideration. Then the open fit tests are made up of others, but for these in particular, we did about 5 parameters. That's immobility, hearing, ambulation, grooming, and time spent executing each behavior. So immobility is freezing time. So the number of time this rat here stops moving, that's basically immobility. So we calculate that period, and that's why it's better when you record. So after recording, another person is analyzing immediately. So you analyze the number of times, stopwatch, then you count that. That's immobility. Then the second is rearing, which is we all know animals, especially rodents, they do a lot of activities to clean up. So that's rearing, try to touch their skin and all of that. So that's, then we also have ambulation, then we have grooming, and time spent in each behavior. Then also we also have walling, where they are trying to touch the walls. Yeah, so that was recorded, but I think we had issues. That's one thing that I myself personally too have learned, that immediately every test that we do today, we must analyze the data that evening. So it's more like work in the lab in the morning, you code or you do your analysis in the evening. It's very much better. So the moment we have to maybe keep it to the next day due to procrastination, one way or the other, maybe you lose a video or you lose the other video, you don't know what might happen. So that's myself I've learned. So, but basically, this was what we have estimated for that, and they have specific function: immobility, freezing time, rearing, ambulation, grooming. Right. So the next is the teammate spontaneous alternation test, and it's basically used to make sure behavior in the animals, especially is mostly used in rodent animals, and this is how it looks like. It's a T-maze. I mean, we must have heard about Y-maze. Y-maze is that of Y, then T-maze is like this. So you see the same dark room, then we have the camera up, then we have the T-maze right at the center. So we have the rats there, which is that white guy in there. So then also is divided into those sections so that you can see it as some divisions, and it's also a kind of a sense. It's sensory awareness. Those lines they help in terms of sensory awareness too. So the test is based on the willingness of the animal to explore the new environment. So they can prefer to go to this arm that's starting from here, go to this arm, or they go back to the arm they came from. So that is that. Then what we did was they were first placed in the start arm of the teammates, and that's this point here. Okay, this point here, that's the start arm, right? So then, okay. Oh, no. All right. Okay. Sorry about that. I was trying to. So from that start time, which I indicated here, then they're allowed to move forward. Then they are allowed to choose which arm they want to go to. So after leaving that arm, they go to the left or the right arm. Then after lot of trials, what we get to see personally when studying them is that they show less tendency to visit the previously visited arm. So it's more like exploration. Yeah, I want to know what is in that place. So when they go, they come back, let me go and check that place. So there's a sequence. So let's assume we have ABC. It's supposed to be ABC, ABC, ABC. So once we have ABC, that's a right alternation. Then once we have, in a scenario where we have ABA, that's already a wrong alternation, right? So those are one of the things that we count. They will also count the frequency of entering new arms, the frequency of entering new arms. So we get to know the number of turns in each arm, then the total duration that is spent in each of the arm. So spontaneous alternation is also used for measuring exploration, and also in some scenarios, memory. So the right alternation or correct alternation is used during memory, but the number of alternations, number of explorations, you can also use that in measuring locomotive function or motor function. All right, so these are what it looks like. Then the last one that we did was the Morris water maze. And what we did was we assessed the spatial learning and memory of the rats. So the truth is, fine, we're studying the cerebellum, but we just need a background understanding of if this actually affected the cerebellum, right? So is it going to affect their memory? And we all know that the memory is majorly initiated, like memory storage, memory retrieval, majorly initiated by the hippocampus and the prefrontal cortex. So, but we just have to check what was the memory state of this, right? So this test was basically to measure spatial learning and memory, and a pool of water measuring about 100 centimeter in diameter and 30 centimeter in depth was used, and an escape platform. And during the training, so the animals were trained 24 hours before the actual test. So what we did is we dip them there. So each rat will be placed in the other three quadrants for a maximum of 60 seconds. That's during the training, to find the escape platform at intervals of 15 minutes between the quadrants until the escape latency period was reduced to less than 50 seconds. So the time which they used to find the escape platform is what we estimate, which is called the escape latency. And it is a very good indicator for memory function and especially learning too. So what we did here to also measure locomotion was to check their locomotive activity. Yeah, how well they can, because in a research, when conducting study, we see a lot of things. Yeah, some things that we might not be able to report. Yeah, you see, although we see some rats that even before the week 2, they already coming up with a lot of motor function impairments, and some of them still survive till even the last day. But by the last day, what you are seeing is completely different. So sorry for that. So by all right. Okay, yeah, sorry for that. All right, so by where was I? Okay, so we measure the escape latency, and one of the things is that there are some things that we see during this neurobehavioral assessment. For example, I was given an example that some animals, they tend to develop this very bad motor function impairments at week 1. So, but one way or another, they survive till the last week. But you yourself, you know that this is very, very, very wrong. So these are the things that we also assess using the Morris water maze. Some of them, the moment you dip them inside the water, trust me, they are gone. They're not even going to put in any effort to get out of the water. So those are the things that we felt the Morris water maze was going to provide us with as information on that. So, and all right. So during the neurobehavioral test, one of the things we also do is physical observation. Yeah, that's one thing that we are taught in animal handling and care, and I feel we should also take into cognizance. Yeah, so try to check if the animals are developing any conditions, and if there's a need, especially when it comes to maybe some rats, extras that you're not making use of, you have the veterinary doctor, you give them to the veterinary doctor so that they can address it. Yeah. But for our administrative groups, you are not expected to give veterinary doctor because I mean, once they administer anything to them, you've spoiled your whole results. So, but some of the things that we saw was oedema, that swelling of paw on the forelip of rats. I hope you can all see that. Then the oedema, swelling of paw on the inlip of rats. So this is below the lid, and it was one of the administrative groups. Yeah, but due to some reasons, we couldn't report it because we didn't. Uh, After deliberation during the publishing with the editor-in-chief, just left it like that. So, but I said I was going to present that here. So these are what it looks like. So the question is, what causes this kind of situation? So sometimes it can be the negative control, and sometimes it can be the environment, right? Some rats just tend to develop this in reaction to, and sometimes it can even be your positive control. I've be engaged in a study where the positive control caused this. So these are some of the things that we also notice because it's important. Yeah, it's important. All right. So one other thing that we did after the neurobehavioral study was that after day 28, we dissected our animals. We had to kill them and remove the brains and the structures that we need personally. So we conducted morphometric analysis. Morphometric analysis is more like measuring the morphometry of these organs. So it can be the brain, it can be the cerebellum. So we measure the brain, that's the total brain weight, and the cerebellum. Then we also conducted the brain volume, but we didn't report it here. We didn't report it here. That was total brain volume. So looking at the volume of the brain. Then basically this was what we did, and it was done using a weighing scale, and we analyzed it using graphical prism. So okay. So this was how it looked like after dissecting the cranial structure of the rats. Then we measured the brain weights on, and we got this averagely these measurements for a control group. And all right. So now for the histological processing and biochemical analysis. So after all of that, the truth is that one thing I've learned also, or that I learned in this study, was that there has to be adequate preparations. In some scenarios, you have a lot of samples and you can't just immediately start working on all your procedures at once. So that's where you need to have a proper storage to be able to keep them, a refrigerator or a fridge to keep the samples. And if possible, you can just try as much as possible um, to keep it properly. So we conducted histological test that's seen the histological architecture of the tissues of the brain. Then we did biochemical analysis that I mentioned, we did neurotransmitter, we did immunohistochemistry. So, and these required that we divided us, that's why we had so much animals, like 10 animals. Although we didn't dissect all of them because, but why we had that 10 was because 10 for each group was that for behavioral study, it is recommended that you have a minimum of five. That's to reduce any, in some scenarios, you might have, what's it called? I've forgotten the name that it's called. I've forgotten the name. This odd number, there will be a particular rat that will just have this increased, yeah. So just to reduce that. So that was why we made use of that. And we didn't dissect all of them. Not all of them was used. Not all of them was used. Some were even, after the whole study, some of them were kept. I had some of them with me, but at the end of the day, some of them later died. But we didn't dissect all of them. So we did histological processing, and basically after the behavioral assessment, morphometric analysis, brain tissues were sized, dissected. Then the coronal sections of the cerebellum were obtained stereotactically, and for each group, 3 rat brain samples were chosen for immunohistochemistry, targeting neon and TNF-alpha expression. So I'll still come to that, what neuron is all about and what TNF-alpha is all about. So then the 3 brain samples per group were used for biochemical analysis. Another 3 brain samples were allocated for histochemistry, that's to assess neurotransmitter. Then the cerebellum structures dissected from the brain samples were embedded in an EDTA sampling battle containing 10% NBF, just to preserve it. This part of the brain was processed to obtain a paraffin wax embedded block for H&A administration. Then the homogeneity of tissue was obtained and centrifuged at 12,000 RPM to obtain the supernatants containing tissue lysates. So after we got the supernatants are the upper part of it, that's the main part. So, and stored at very low temperature, GPS and other assays were conducted using it. So now, back to what we have here, which is immunohistochemistry. So immunohistochemistry is just basically you looking at antigen to antibody reaction. We want to understand, okay, let's take, for example, you call this person, you said this person has this disease. Let's take, for example, Alzheimer's disease now. So there are some proteins that you expect to see that are expected to be expressed in the brain. And examples of them are, this is supposed to be an interactive discussion. I would have loved if we could contribute to this, but maybe after the class, we are still going to have a very, very interactive discussion, not a class, a session, after the session. So one thing is, those proteins that are being spread, an example of them is amyloid beta, for example, and we also have tau protein for Alzheimer's disease. So each of these proteins, they are antibody to antigen reaction that you can use to examine it, right? And also, there are also some also some antibodies that are also present that can be used to visualize these proteins. Let's say, for example, now neon. Neon is used to understand the number of matured neurons that in that brain sample. So in a scenario where you have a lower proportion of matured neurons, that means the neurons are not maturing, right? So if those are one of the things, and you need to compare with other studies to be able to generate your findings. The TNF-alpha, that's also used to check if there are some level of inflammation, right? So, and that was what the immunohistochemistry was supposed to do. With histology, you're going to see the histological architecture. You see how, okay, this neuron is damaged, the cell body, this and that. But to have a better interpretation of what you are seeing, that's why we conducted the immunohistochemistry. All right. So for the immunohistochemistry procedure,
recording in progress.
all right. So for the immunohistochemistry procedure, what we did was immunohistochemistry is basically you trying to identify antigen to antibody reaction, basically, like I explained. So the cerebellum was fused in a 10% NBF, then we dehydrated and embedded in paraffin wax, just like we did for the H&E. So we had to do sectioning. That's one stage of immunohistochemistry, and they were cut into charged and rehydrated. So after that, antigen retrieval was conducted, then we blocked, then we use the anti-neon antibody at a specific proportion, right? And one thing I've learned so far about immunohistochemistry is that there has to be a proportion. The proportion that I use for 7 samples might not work for when I'm working with about 20 samples. So the more samples you have to increase it. And also, these antibodies, sometimes you can use it twice, 3 times, that you can recycle them. So in scenarios where this is the third time you are using it, sometimes you might need to increase the proportion, because the more you use it, the lower its viability or functionality. Let me just put it like that. So, and seconds were then incubated in horseradish peroxidase micropolymer goats and we use anti-rabbits. All right. So I don't know if we are all like me, but me, one of the questions I've always asked when I see things like anti-goats, anti-rabbits, and I'll be like, we are making use of rats, right? So for rats, for specific antibodies, it's recommended that you use maybe anti-rabbit, anti-goat. Like there are specific antibodies that you use for that. And that's based on what you read online. You check which particular antibody was used. Because if rats, for example, you might not get the expression that you expect. So those are the things that, but for these, we use anti-rabbits for it. Then for TNF-alpha, we measured it, that's the tumor necrosis factor, and using the assay kit, then through the Immunite 1000 system, according to the manufacturer's instructions, the detection limit of the kit we obtained from the manufacturers, and at the detection limit of 1.7 over 1000 mg per microliter. All right. So one thing I said was that we're going to do a step-by-step protocol for the immunohistochemistry, and that's what I'll be going through now. So immunohistochemistry, like I said, it looks like a very complex procedure, especially when you've not done it. And I personally, I would have loved that I would do a kind of video, maybe if I was in the lab, do a video of each of the procedures so we can do it together and just run through together. But we are currently in the festive period. It seems like we are in a we didn't schedule it when we were supposed to schedule it. So, and the lab is locked now, but I'll just take it one by one so we can go through it. So after it has been deparfuranized, it has been blocked, H&E and everything, then we now have the block, right? So it has been sectioned. Let's not forget it has been sectioned, tissue processing and all of that. Then we now have it on the slide. So the first thing is we try to rehydrate the slide. And rehydration of the slide, this procedure I'm sharing here, I'll share it to your chapter head, so that for anyone who is ready to try it out. Sorry, I got a notification. That's why I'm kind of distracted. Sorry for that. All right. So the procedure basically is what we use at the lab, and that's why I'm okay with sharing it. All right. So the first stage is rehydration. So rehydration is basically we put it in saline for 10 minutes. So it's more like we have the bottles 1, 2, 3, 4. So saline 10 minutes. After 10 minutes, you move it into 100% ethanol. Then you move it to 95% ethanol. Then you move it to 70% ethanol. So it's timed 10 minutes, 5 minutes, 2 minutes, 2 minutes, 2 minutes. So why are we rehydrating? We are trying to make the slide, the tissue more rehydrated. Yeah, so after the old tissue processing, some of the tissues might have been dehydrated. Yeah, we all know about the rigorous processing of tissue processing. So we need to allow the tissues to come out again, and also the cells to become more rehydrated. So after that, we input into distilled water for 2 minutes. So the next stage is the antigen retriever, which we prepare the antigen retriever solution, and using the following component, that's citric acid or masking solution, that's the brown solution stored in the fridge. Actually, we actually stored in the fridge here. So that's why it's like that. Then distilled water, that's 213 ml. Then we prepare that. So then we boil. So it's going to be boiled for about, I think, 30 minutes. Yeah, for 30 minutes. So what the antigen retriever does is that it's trying to make the antigen to become retrieved, just from the name, become exposed, so that for easy antibody to antigen binding, basically. So we are making the antigen more exposed, more retrieved, so that it can allow for more binding. So then after that, we block or wash. So we place the slide on our flapping disc, yeah, which we use for flapping. Then we draw a circle on each slide, then we apply PBS or TBS, as the case may be. Then we that's we prepare a hydrogen peroxide solution with the component hydrogen peroxide then PBS 5 ml, then we repeat this preparation 5 times. So it's more like you apply PBS, like after you apply the and PBS of 5 ml, you apply it, then you flap, then you now apply PBS 5 ml, 5 ml, like 3 times. So the next stage is the antibody application. And I feel like after this stage, this is where it takes a lot of time because you have to wait for like 2 hours. So you get the antibody after the hydrogen peroxide treatment, and you remove the PBS. Basically is just to remove any remnants of the hydrogen peroxide that is still remaining on the tissue, right? So then we prepare the antibody at a dilution of your proportion. So I mentioned the other time we used 1,000 ratio 1,000, 1 ratio 1,000 for neon. Then for 10 alpha, I think we use 1 ratio 1,5. All right. So same thing for this 1 ratio 1,5. If that's the proportion you are making use of, then we prepare that, then we administer it, or we use it, apply it on the slide. So after the primary antibody, we wait for about 2 hours. Then we now apply the secondary antibody. So most times the primary antibody, you can take it back if you care. You can take it back, but after taking it, you wait for you apply the secondary antibody, then 1 drop per slide. Then you wait for about 30 minutes. So after this step, you watch for PBS for 5 minutes, repeating this step twice, right? So then you apply your DAP. So the DAP is a stage where you need to ensure that everything is on place, because you are trying to, as much as you already have information of, okay, this is the number of time you are expected to wait before you can remove the DAP solution. So what DAP does is that it allows your antibody to antigen reaction to be expressed. That's what DAP does. So once you want to apply DAP, just get your stopwatch with you, and you also get your microscope and your system connected so you can see the reaction. And it's a very interesting reaction when you are seeing the DAP expression, because something that is just plain, gradually you are seeing the neurons expressing, you are seeing the brown expression gradually. So that's that. Then after applying the DAP, you stain, then you have to dehydrate. Rehydration comes from xylene to 100% to 95% to 70%. I mean to 90% to 75%. Why dehydration comes with 75, 90, 100. So rehydration is coming up, down, while dehydration is coming down, up, right? So then we wash with xylene, then we mount. So we use mountants for mountants. And one of the procedures we should take note of is let's ensure that we engage in the laboratory precautions, and that's make use of nose mask, gloves, because some chemicals like xylene mountants, they are very carcinogenic. And I mean, if you place your hand gloves inside a xylene bottle, xylene concentrated bottle, you see how it's going to melt in a couple of times. So if xylene is touching your skin, then you know the kind of damage that it's doing. Although it might not just affect it once, but after a lot of time to affect it. So that was one of the procedures. That was the immunohistochemistry procedure that we used for this study. And the next stage was the glutamate assessment, where we assess the glutamate level. And basically, the brain tissue was deproteinized based on a method that we used, and fibrin were rapidly weighed and grounded to a powder in a stainless steel tissue grinder called a liquid nitrogen. Then 500 microliters of nitrogen SO4 were frozen and grounded in a separate stainless steel tissue. Then it was now called liquid nitrogen. So the two frozen powders were now combined into a plastic centrifugal tube and allowed to warm for 4 degrees Celsius. So these procedures are basically, they are assays, right? They are assays, and there are specific methods that one will just need to adhere with. And after the whole procedure, it was based on the spectrophometric assay, where we have the reduction of 3-acetylpyridine adenine diclonotide that's applied to APDH. So that was how we were able to get the glutamate concentration. So next is GABA, that's the gamma aminobutyric acid, and it's basically based on the assay of the competitive inhibition enzyme immunoassay technique. So the standard samples were added to the microliter plate wells with a Biotin conjugated antibody that is specific to GABA. So avidin conjugated to horseradish peroxidate was added to each microplate well and incubated. So after the solution was added, that's the TMB, we added sulfuric acid solution to terminate that reaction. And the range was measured using spectrophometry at a wavelength of 450 nm plus or minus 10 nm. So basically, I will just run through the preparation of the tissue homogenates. The tissues were rinsed in ice cold PBS to remove excess blood thoroughly and weighed before homogenization. Then after that, it was minced to small pieces and homogenized in fresh lysis buffer. And the resulting suspension was sonicated, used as sonicator with a ultrasonic cell disruptor. Then the homogenate was centrifuged for 5 minutes at 1,000 times gram. So the GABA procedure, this was the procedure that we followed. This is a detailed information on the procedure that we followed. But due to time, what I would do is it's not even on the manuscript that we published, this particular procedure. It was when I was going through there and I had to go and look for it, and I said I was going to share it. But regardless, I will share everything in the document, or maybe when I share the PowerPoint, you can always get it from here because of time. So then we examined glutathione peroxidase and glutathione reductase activity, and the formula for this after the whole procedure is that the glutathione reductase consumed is equal to the 245.84 minus glutathione reductase remaining. So the glutathione peroxidase activity is equal to glutathione reductase consumed over the milligram protein that was used. And that was the procedure that we followed. Then we have the photomicrography and data analysis. So the photomicrography of the slides obtained from histological and immunochemical processing of tissue were captured using binocular microscope, and we connected it to a 5.0 megapixel camera. Then statistical analysis was conducted using the GraphPad, and the results generated were analyzed using ImageJ software, especially for the immunohistochemistry, while the glutathione reductase, glutathione peroxidase, glutamate, GABA, they were analyzed using GraphPad. So we use one-way ANOVA Turkey test to determine any significant difference among the group, the confidence limit of 95%. So, okay. So what we'll be doing now is a practical section on the quantification of immunohistochemistry images using ImageJ, and that will last for about 15 minutes. I think we started the session 7 o'clock. We're going to end 9 o'clock. So we'll be doing that for 15 to 20 minutes. Yeah. So please, if you have all right. So if you have ImageJ installed on your system, and maybe you want to just engage with us as we continue, you can just indicate so I can quickly send the documents, the data to you so we can use it together. But if you don't have, I'm going to share my screen right now so that we can go through it together. All right, so just give me a minute. Let's just hold on. And also, if there are any questions we want to ask, we are going to take them. I think that part is going to last for about 20 minutes. So you can just note it down. It's going to be an interactive section where we can discuss together. So just give me a minute so I can share my laptop screen. All right. Thank you for waiting. Sorry for the delay. Just wanted to share my screen. Okay. Let me just share my screen completely. I think that's much better. All right. Okay, can we all see my screen, please?
I think I can't see anything yet.
Okay, you can't see my screen.
It might be like loading or something. I'm not sure. It's right now it just says that started screen sharing, but it's like a black background.
Okay. Okay. What about now?
I still don't see it. Oh wait, I see it now. I see it now.
Okay, all right, all right, all right, all right. So basically, I believe we are all familiar with how if we are not all familiar, at least some of us understand how immunohistochemistry images are like. So they are like our normal pictures that we have where we save them, and they are taken in different magnifications. We have the times 40, we have the times 100, then we have the times 400. So the times 40 is basically for you to understand, okay, this is how everything looks like. Then we have the times 100 to look at it at a more visible level where you can just see the arrangements or the architecture. Yeah. While the times 400 is to see the specific neurons, yeah, how they look like, the nerve cells, how they look like. All right. So we'll just be going through it now and seeing what we can do with it. All right. So here we have IBA1 actually. So the part about this study is we did a lot of other, aside the TNF-alpha and the neon, we conducted IBA1 quantification, and we also conducted tau. Yeah, tau. But tau was focused on the prefrontal cortex area. We're just trying to get as much findings that we can. Yeah, although still in that scope of the study. So, but for the published material that we have out there, we only reported that of the neon and the TNF alpha because that was majorly what the cerebellum what we wanted to see. Yeah. So and for the IBA1 and the I think the IBA1 and has been published in another area. Yeah, it has been published in another area. So, but I'll be running through using the IBA1 as a case study now. And all right. So let's assume we have this image J. This image J that we have here is this guy that looks like, I don't really know what I can call it, but it's a golden something. So what you do is once you have it installed, you open the file, you can just do kind of a copy. Yeah, maybe let's assume you have it here. Then you just raise it up and leave it here. So automatically what it does is that it copies it. It copies it there. Then you can now make use of other analytical tools that you want to make use of. So in Imager, you have the file option where you upload images and everything that you want to do. Then we have the edits, right? We have the edits where you can make necessary edit that you want to do. Then we have the image. Then we have the process. That's you want to smoothing, you want to sharpen. I've uploaded the image already
and we have the analyze, where you can do some other analysis that might be necessary. Okay. So the process is basically, maybe you want to add shadows, you want to also do image calculator. Yeah, you can do some calculations on it. Then we have plugins. The plugins is what makes, it's like an extension, just like we have extension in Word document. So where we have other functions of the software as it is. So the name is ImageJ. Yeah. So then we have the macros, utilities, tools, Bezier curve tool. So, but it depends on the function that you want to make use of. Then we have scripts, Python animation, rotating polygon, and all of that. Then 3D image is also used for 3D surface plots. So, but I won't go into that today. I'm just trying to, for those who are just new to it, just to let you know what the functionalities of what you can make use of it for. So then for the analyze, that's where we now have the cell counter. So the cell counter is used when you want to count the number of cells, and that's what basically we'll be doing today. Then we now have the immunoratio. The immunoratio. Okay. Then we'll have the immunoratio. So for today, we'll be making use of the cell counts. The cell count, so that we can count the cells and know the number of neuronal cells that are present, especially when it comes to things like neon. Neon is basically you understanding the number of matured neurons that are present in that cell, in that slide. Yeah, in that slide. So, and don't forget that during immunochemistry, we ensure that we are not just taking every part of the brain. That's why we do sectioning. So we have the cerebellum part, we have different parts like that. So for the cerebellum, you don't count, like, I mean, you don't take two duplicate images, then you count it together. So you need to ensure that there are different areas of the slides that you are counting. So now I've uploaded one, and I think we have it here already. I've uploaded one. This is it now. All right. So this is what we have now. I've uploaded one of the pictures, and I don't know if that's clear enough, but to my understanding, it's quite clear. So this is IBA1. This is IBA1, and for what we can see here, we can see variety of cells that are, some are closely packed, some are beginning to disintegrate. Then you can also see some brownish structures. So you knowing what you're expected to see is also based on an understanding of some research that you do, because for example, you might be expecting to see this, right? And in some scenarios, all your groups, you are not even seeing anything related like that. So you need to understand what that antibody and antigen reaction is about to produce, then what you are expected to analyze. All right. So what we did was that for neon cells, we counted the number of the matured neurons, and the matured neurons tend to appear brown. They tend to appear brown. So we counted it. So once you have this cursor here, what you do is you click on it. Okay. Coming. I think I've not installed the cell counter now. Okay. So now let's go back to it. All right. So you click on it, it gives you this. All right. So let's assume you click on this now, for example. So you initialize, that's the first thing. So we have different types. We have type 1, type 2, type 4, as the case may be, anyone that interests you. But the whole thing about this type is you being able to know the number of photomicrographs that you're counting. So we have type 1, type 2, type 3, type 4. You need to initialize first. So, but the first stage is you initialize first. Then now you select the type that you want to we want to use. So let's assume we are starting with type 1 because this is the first photomicrograph that we are trying to count. So this is the brown. One, 2, 3, 4, 5, 6, 7. So don't forget that in photomicrograph histology, we have things called artifacts. Sometimes we have tissues that overlap, and we also have overexpression of what we expect to see. So those are some of the things that we take note. So for example, now this now, and sometimes you might need to edit the image, increase the contrast so that you can see what you need to see. So those are some of the things there. So these. I don't know if we are following, but I think we can see them. 1, 2, 3, 4. Okay. So it's called cell counter. We are basically counting the cells that we want to subtract out. So that won't take a number over time. That's for type 1. So what we do is after there, we add, then we save markers. So that's type 1. So the next thing is to upload the next image. So we already have that image now. We save it. So the next thing is to upload the next image that we want to count. Then we save it as type 2. So at the end of the day, don't forget we are in a particular group. At the end of the day, we sum all the cells together. Then we get the number of the particular antibody that are present in that group. So now, let's assume for group 4, I think we already got to 33. Let's assume we have about 3 of them now, or 4 of them now. Yeah, we now have about 33 times 4. Let's say that's about 100 thereabouts. 120, let's say that's about 120 thereabouts now. So for group 4, we have 120. So for group 1, we reported maybe 50. For group 2, we reported 100 like that. So that's what we'll now take to GraphPad to go and analyze. So that's basically how it works. And I think I have this version. And for those who might be interested, you can just reach out to your chapter head or the organizer of this. I would upload a Google Drive link of the image there. It might be useful for you maybe later on. And also there are a lot of other functions. Like I said, there's what we call immunoratio. Immunoratio. Yeah, immunoratio is just uploading the image, then to give you a percentage of the brown structures or distinctive neuronal cells or structures in that tissue. So that's what immunoratio does. To give you the number in form of percentage or proportion, so you can utilize that during your analysis. So, but it's specific too, like I said, for neon, you do cell counts. I know immunoratio, you do it for things like GFAP. You can make use of immunoratio for GFAP. For things like yeah, basically, but you need to be careful too of which tool to use for which particular antibody to antigen reaction. All right. So that's that about the cell counter. They will also have grid. Let's assume you want to make a kind of a grid. Maybe the image is so big, like you have a lot of expression, and you're just trying to ensure that you do it, take your time to ensure that there are no errors. Yeah. So you can include a kind of a grid so that once you are done with this grid here, let's say you're able to identify one. This was some of the things that helped me personally. I move to the next. So I ensure that I take every part and I do not come back to the ones that I've done before. Although there's going to be an identification basically, but those are one of the things that we take note of. So Imager is a very good software, and I personally would recommend for anyone who is interested in working on immunohistochemistry so that you can get a proper analysis of your results. All right. So then if you want to increase the contrast, like I mentioned earlier, in some scenarios, the contrast image is not, you cannot see the neurons as much as you can. Yeah. although you really need to be careful too to ensure that you don't use those images when submitting for presentation or for publication thereabout, because it means that you've modified the neurons, you've modified the whole original thing. But in cell counting, so that you can know what you are seeing, you might need to increase the contrast so that you can know what you are seeing. So those are some of the things, and I believe it's going to be very useful for some of us that have not been able to use it. So that was what we used for our study. We did cell counts for neon, and for TNF-alpha, I think we did immunoratio. Yeah, and that was how we were able to get our study. So I'll be moving to the next, which is GraphPad analysis now. All right. So this is how GraphPad looks like. GraphPad is a software that we use for analysis, just like we have SPSS, that's statistical package for social sciences. We have R, we have Python, different. But GraphPad does not require coding. It's just basically you inputting your data, you analyze, you get your findings, and you plot graphs. So this is how the interface looks like. And I think you need a premium version to be able to use it. Although some people make use of patched version. Yeah, but you need to subscribe to be able to make use of it. So let's assume we are starting a new project. So we have file, we have edit, we have view, we have insert, we have change, arrange, family, window, help. So this file, you have all those options: new, open, close, or save, save as, and all of that. So these are some of the history of the projects that we've done. They are automatically going to be saved. All right. So let's assume we are starting a new project now. That's a new project file, new. New project. All right. So this is the interface you're going to get. You're going to get SY, you're going to get column, you're going to get group, you're going to get contingency, survival, part of whole, multiple variables, nested, like that, like that. All right. So these options, they are basically you knowing what you want to compare. And like we know, statistics is not just you playing around. You need to know what you want to compare right from onset, what you expect, what you want to see, what you want to project, and also knowing the particular analysis that is okay for this. So now we said we use one-way ANOVA. There's two-way ANOVA. Why are we not making use of two-way ANOVA? So those are some of the things, some of the questions. We have t-test, we have f-test, we have a number of them. And also during one-way ANOVA too, you have to do comparison. We have things like Turkey, they call it post-hoc test. So those tests, knowing the particular test that I expect to do even at that stage is also important. So that's why some basic knowledge of statistics is very important when we are doing our research. So during our own analysis, we made use of grouped, and we selected enter, that's replicate side by side columns. Then and why did we use group? We use group because we had like 5 animals in a group. I think for neurobehavior, we had 5 animals. For immunochemistry, we have, I think 4. Yeah, 4. So those are the things that we are trying to ensure that we stick with. So we create this, then you have something like this. Yeah, you have something like this. So we name the groups. Let's assume we have group group A, then we have group B, then we have group C, then we have group D, just to ensure that we have everything set up. So group A, group B, group C, group D, then let's assume we have 5 animals in one group, then we have the title. So assuming we are comparing, like, let's say we have in a particular group now, we have male, we have female, that's where this will come into place, something like this. But since we are not making use of that, we just let that slide. All right. So let's assume for group A now, for our immunohistochemistry, we had, for the first one, we had 120. The second, we had 122. The third, we had maybe 101. The fourth, we had 106. And the fifth, we had 105. Sorry, this is just a tutorial. It's nothing serious. And so if we do not get something quite sensible or something that's awkward, don't blame me, please. I'm just trying to simulate something that we can use here. So let's say for group B, we have something like 99, we have 88, we have 66, we have 55, then we have 34, 32 rather. Then for group 3, we have maybe 150, we have 120, we have 140, we have 160, and we have 180. In group D, we have 102, then we have 103, we have 104, 104, then we have 112, then we have 116. All right. So we have group 1, group 2, group 3, group D. Then you can already start the project info if you care. But yeah, there's that option for those who can just immunohistochemistry. How you see beyond. All right. So then we do the analysis now. So here we have different analytical tools, or should I say methods, that we can use. We have column analysis, XY analysis, and so on and so forth. So we said we're going to use one-way ANOVA, right? And why are we using one-way ANOVA? One-way ANOVA is a form of a more extensive non-parametric test that we use when we are comparing different groups and different duration. So, but for two-way ANOVA is more sophisticated for my own understanding. Okay, let's assume now we have, okay, I don't want to make use of, we are not doing immunohistochemistry, let's do neurobehavioral. Yeah, okay, let's make this immunohistochemistry, then we'll do neurobehavioral after. All right, so column analysis, one-way ANOVA, group A, group B, group C, group D. Then we have multiple comparison, compare the mean of one column. Okay, let me not rush this. So once you click on any neuroparametric test, you have parameters. So you expect to select which one works for you. So we have no matching appearing, each row represented match or repeated mirrors data. So here you just leave it as no matching. We are not trying to pair anything here. Pairing means that you are trying to pair one group with the other. So we are not pairing anything, we just leave it there. Then we are using ANOVA. We are not using non-parametric tests. We are using ANOVA. So then assume equal S this, use ordinary ANOVA test. So there are other forms of ANOVA like Brown-Forster, and we also have wedge ANOVA. So, but we are just going to make use of the ordinary ANOVA test. So they are basically modifications of the ANOVA that we have. Then we have the multiple comparison. Repeated measures, since we are not doing two-way ANOVA, we're less likely going to make use of repeated measures. So we have multiple comparison. We are trying to compare the mean of each column with the mean of every other column. So for neurobehavioral, that's when we get to do comparing different groups on different days. So then the next is options. Use so GraphPad gives recommendation. You can use Stocky, Boffereni, CDAC, CDAC, Newman. So we use Stocky. Then you just leave all of this. Multiple comparison options, family-wide significance, leave it at 0.05. If you want to use 90% confidence interval, that's left to you. But I personally, most times I use 95% confidence interval. So graph confidence intervals, then I turn on my descriptive statistics. I want to see the mean median mode. I want to see the standard deviation. I want to see the standard error of measurement. So I turn it on. Then I show significant values of show significant values. So now we have APA, we have NEGM, we have GP. So in scenarios where we are looking at 0.05, 0.01, 0.001, we can just make use of this APA. This for me is the closest. GP is more extensive in the sense that take this for example now. We have the 0.05 starting from, that's the closest, starting from 0.0332. And in this scenario, we get to see decimals. And me, I really am just okay with getting, if my values are 0.05, I should just get like 2 to 3 decimal points. But the moment I'm getting like 4, 5, it becomes more complex. Although it's important, the numbers behind are important, but I prefer it approximated. Yeah, and that's why I go with this. and residuals, okay. And that was basically what was done for the immunohistochemistry. So after then, you get your results. So this is the p-values, that summary 0.01, the R squared. So one thing, I think one of the discussions we've had most times in the lab is this p-value thing. Is it 0.01? If there's no 0.01, does that mean our results are not correct? No, that's absolutely wrong. The fact that you did not get 0.01 does not mean or you not get a value that is less than 0.05 does not mean your studies doesn't have any value. So basically, sometimes the p-value does not really define our results. You can check other variables like the R square. You can check other variables like increase, like reduction. Those mean standard deviation. They can be a pointer to a lot of information that one might be ignoring. So we have the multiple comparisons here, group A versus group B. Don't forget it's a random figure. It's not our study, so let's not take it too serious. So we have group A versus group B. We have a significant difference at 0.01. So I would, for those who are not aware, 0.05 p-value, we have 0.05, 0.01, then we have 0.001, then we have 0.001. So p less than 0.05 is 1 star for me. Then p less than 0.01 is 2 star. Then p less than 0.001, that's 3 star. Then p less than 0.0001, that's 4 star. So that way, those are the things that we use to indicate on our plots. So we have this result now. We have our descriptive statistics here. We have the minimum, the 25% percentage, the median, the maximum, standard deviation, standard error of measurements. Please, we can just note our questions. It's something that we can always open again during the interactive section. So note your questions down regarding this analysis and everything. I just want to ensure that we meet up to time, and that's why I'm just trying to rush through it. So after that, GraphPad automatically plots the graph for you, basically. So this is what we have now. And as you can see, but I like to make it more designed and well designed well enough. Yeah, I like to design it properly. All right. So then you can do a lot of other formatting to ensure that the color is quite okay enough. And we have colors. I prefer this color basically. So now reporting the plot is basically based on what your study did. So I mentioned the other time something around 0.05. That's p-value of 1 star between 0.05 less than 0.05. And that's group A versus group C. So let's assume group A versus group C now. We can just do this to create a kind of let's make use of this. All right. Group A versus group C now, 1 star. So we just indicate it there. Then based on our other one, which is 2 star, that's group A versus group B at 0.09, can also indicate it there like that. All right. So basically, you don't forget that you have to put your X and Y axis. So let's assume this immunohistochemistry. So just do expression of neon. Then this will be administrative groups. X and Y axis is something that is very, very important during analysis. So after you're done, you can just copy the graph and you paste it on PowerPoint. So you have your study. So that was how we came about some of the graphs that we're going to be seeing in a couple of times from now. So I'm going to go back to my screen now so that we can go through it together. So that was how we did the analysis basically. It was just basically for immunohistochemistry, quantifying the data, bringing it into GraphPad. For neurobehavioral data, I said that after the conduction of the neurobehavioral test, we collect the data, we analyze that same day. So there's this quote, experimental lab in the day, then the computational lab at night. So you perform the lab activities during the day, and at night you analyze everything that you got during the day. So because if you wait till the next day, sometimes procrastination. Everyone, me myself, I made a very good example of how one of the recordings of important parameter we missed it due to procrastination. So I personally have learned my lesson, and I ensure that everything that I do that day, I ensure it can be stressful, but analyze it that same day, so you know that's gone. And so that if there's any error that you find out, you can conduct it again the next day. It's not too far. It's not too far. I mean, we are supposed to do a study, neurobehavioral study, day 7. For our own, we did it day 7, day 3 for wire hanging. Then the other test, day 7, day 14, and day 28. So if there was an error on day 7, we were able to correct it day 8. But the moment you don't correct it and you've gone far to maybe day 12, you can't do it again. It's gone. So those are the things that we take into cognizance. So I'm going to be going back to our PowerPoint presentation now. And time is fast spent, so we'll just run through it. But regardless, all right. Let me share my screen. All right. So we've done that of graph part prison. Then these are the results now. So what we got was that for the body weight, there was really no significant difference, but we could just see this control, the Phyllanthus Amarus group, where we administered just Phyllanthus Amarus only. Yeah, the thing is, we call it positive control, but we really cannot say. Sometimes your positive control can turn to a negative control. So we need to know, okay, what is the particular effect of this particular extract? Yeah, this particular extract. And I forgot to mention that it was done through aqueous extraction. But one way we are looking at for future direction now is doing molecular docking to identify the particular compound that is responsible for that function. Then we administer it to the rats. Then we also conduct it on cell line. And that's the part we are working in now. Yeah, one of the parts we are working in now. So, but I will just, for the sake of this presentation, this is what I'm going to be presenting. So then the Phyllanthus Amarus, then the potassium dichromate, then the DPZ, that's the donepezil. Donepezil is a standard drug, actually for memory. But we also saw that in some scenarios, it helps as neuroprotection. Yeah, so and that's why we made use of it. Then we have high Phyllanthus plus the potassium dichromate, the low Phyllanthus plus the potassium dichromate, then the donepezil plus the potassium dichromate. So for the last group, which is donepezil plus potassium dichromate, there was a significant increase in their body weight, better than even our normal control. We really do not know why that happened, but that was our result, and we had to release it like that. But we saw a reduction in the potassium dichromate only group. In fact, I can say personally, because I worked with the animals and I saw how some of them were dying. They were dying, they were losing weight, they were coming up with a lot of conditions. Yeah. And so for the brain weight, we only saw a significant reduction in the brain of the potassium dichromate, and we saw a significant reduction in the cerebellar weight of the potassium dichromate. Then the donepezil only, the brain was intact. It was even significantly more increased compared to the potassium dichromate group. So just like we know that donepezil is very, very good in protecting against memory, I think it's one of the few FDA approved drugs that we have against for managing Alzheimer's disease patients, right? So, but for our study here, we're able to see that it's maintained the cerebellar structure. So that's our own finding, and that's what we are just asserting. So then we, like I said, we did a couple of neurobehavioral tests, including number of crossings, number of rearing, time spent in theta square, frequency of entries into theta square, the vocabulary, the freezing time. So the number of crossings, we saw a significant reduction. It was a progressive one in the number of crossings of the rats. Like I said, the potassium dichromate, they were becoming very weak. You put them in the test equipment, and you will see them, some of them will just stand, just looking. Some might even slip off right there. I don't know if that's the very deleterious effect of the potassium dichromate. And that, even as a person, teaches me that this air that we breathe, if so much chromium are inside it, and although it's kind of reduced, but in some areas, industrial area where we have these chemicals that are very present, if this chemical can cause these deleterious effects at this progressive and fast rate in animals, what about we humans? So we were able to see that progressive deterioration, day 7, day 14, day 21. The number of rearing, same thing, day 14, day 28. Then for the time spent in zeta square, there was no significant differences. Then for the frequency of entries in zeta square, no significant differences. But one of the things that we could see was as at day 28 of the frequency of entry in zeta squares, the potassium dichromate group was not even going to zeta square at all. So the number of crossings, number of line crossing, number of rearing, they are basically movement activities, right? And they are locomotion. And frequency of entry in zeta square is also to test anxiety. Zeta square is we have a center in between the box that the rat is ready to go into if you are not faced with anxiety or But on the other hand, to counteract that, what we saw was depression. The rats were not moving. Some of them were not moving. Some of them would just move a bit, they will rest. So those were some of the things that we were seeing. Then the fecal bulli, that's the sheets of the rats. Yeah, let me put it like that so that we can understand. So we counted them, then we saw that result, and that's what we have here. Then it was kind of very much increased for the potassium dichromate and the donepezil plus potassium dichromate. Then for the freezing time, that's the movements. As at day 28, the rats were almost not moving. That's just the word to say. They were almost not moving. Just few of them. Some of them already had some conditions that they were battling with, but they tried. I mean, at the end of the study, even during the study, we had to name each of the rats different names of we group in our group because we were feeling for them. Yeah, we were feeling for them. So that was day 28. Then for the time spent in holding the wire, that's for the wire hanging, and the frequency of T-maze, that's our finding. We saw a significant reduction on day 28 for the frequency of entry in T-maze. Then we also saw a significant increase in escape latency. Yeah, in the rat. It took more time before they could escape from that Morris water maze. Then we saw the time spent in holding the wire. The potassium dichromates were immediately they held the wire in about 3 seconds, they were down. That was basically a summary of our results. Then for the Phyllanthus Amarus, it was very, very, I don't really know how to describe that, but we saw a very potent effect on it. So this in particular is what interests us to go into seeing the particular compound that is responsible for this. So, and also seeing how we can bind and do some molecular docking with other neurotoxic agents to see which properties and which pathway are responsible for this. So then these are the H&E images. We could see cerebellar histology. Then we saw neurodegenerative changes in some of the potassium dichromate group as indicated. Then some of the neuronal structures in some of the groups, the high and low, were still okay. But for the those that were administered PDCh, we could see degeneration. Yeah, we could see also some vacuolation in between. Yeah. Then for the immunohistochemistry, this was how after the photomicrograph, we quantified it and we saw a significant increase in the donepezil group down to the donepezil Amarus and the low Phyllanthus Amarus compared to the potassium dichromate only group. We saw a significant potassium dichromate neon expression compared to control. So that's just to tell us that neon expression was very much reduced in the potassium dichromate group. So then the same thing for the TNF-alpha, but it was kind of different in the sense that for the potassium dichromate and the donepezil plus potassium dichromate, it was significantly reduced potassium dichromate group. We really do not know why that happened, why the donepezil group had so very much expressed TNF-alpha in the cerebellum. And those are the things we are trying to look into. Yeah, and I believe that with time we'll understand more because the potassium dichromate too, it was highly expressed too. So it was also expressed in the positive group, that's the donepezil only group, and it was also expressed in donepezil plus potassium dichromate group. And that's what we have there. So we conducted glutamate assessments and GABA assessments, and in glutamate, then a significant increase in GABA level. And what this means is that the neurotransmitter pathway that is responsible for motor coordination, motor function, and various movements of the body are being affected. Yeah, the transmission is being affected. Then the glutathione reductase and the reduced glutathione, which are potassium dichromate group, and all of that. So basically, our key findings: rat treated with Phyllanthus Amarus for 28 days showed no abnormal behavioral changes while handling movement and brain structure. And brain structure examination revealed no edema or inflammatory changes. Then AP treatment protected against weight loss induced by potassium dichromate, with the AP treated group maintaining normal body weight throughout the study. Then AP treatment significantly improved the time spent hanging on the wire in rats treated with potassium dichromate, similar to the effect of that donepezil, just like reported in the results. Then in the open field task, Phyllanthus Amarus treatment improved the locomotion and exploratory behavior in potassium dichromate treated rats, similar improvements observed with donepezil. Then the Phyllanthus Amarus treated attenuated potassium dichromate induced neurotoxicity and maintained regular expression of neuronal nuclear protein and exhibited a protective effect against the inflammatory factor TNF-alpha in the cerebellum. And for conclusion, this study provides behavioral, histological, immunochemical, and neurotransmitter findings that the aqueous extract of Phyllanthus Amarus leaf extract is anti-inflammatory and significantly attenuate neurotoxic effect of potassium dichromate, which is also chromium, by mitigating locomotion, posture, and coordination impairment. So we appreciate the Laboratory for Experimental and Translational Neurobiology, University of Medical Sciences, Ondo State, which was formerly known as Neurolab FUTA, because we conducted the laboratory study there at FUTA, but it has now moved to University of Medical Sciences, which I'm privileged to work in as an intern, and I've completed my internship experience there, but I still come around to engage in some studies, yeah, where necessary. So we appreciate them for technical support in providing the necessary equipment, reagents that we did for the laboratory studies. And these are my references. Thank you very much. So we have come towards the end of the session, and it's time for interactive question and answer time. So I believe we have questions and answers that I wanted to ask. This is the time for you. This is 8:58, 8:59. Due to the time, we did not start early, so I would appreciate that you give us about 5 minutes extra so that we can discuss. All right. So the call is open now for questions. If you have any question and any addition or should I say constructive remarks, yeah, it's very much open so that we can discuss, because that's why we are here. We are not here to me just sharing knowledge. It's supposed to be an interactive discussion. But I didn't mean we are allowing interactive discussion, we will not be able to complete the session, right? So it's now open for question and answers. Thank you very much.
Thank you. I had a question about like the test with the mazes. So like did you guys test each week for like have the rats do the maze? Because like if you do once like the rats go through the maze like multiple times, do they like start to learn how to do the maze like faster? And does that affect your results?
Sorry, please, can you come again? Sorry, I didn't get that clear. I think network issues, please. Sorry, can you come again?
Yeah, I can type it out.
Okay, right. In the meantime, if you have other questions too, we can ask. Okay, all right. Thank you very much for this question. It's a very brilliant one. So for the maze, we do not train all the rats for all the maze. We have T-maze, we have Maurice water maze, we have the open feed test. The only one that undergoes training is the Maurice water maze, and that's based on the protocol. That's based on the protocol, because if we just start our neurobehavioral test and we just place the animal or the rodents in the Maurice water test equipment, some of them might just drown immediately, right? So, and Maurice water maze is quite different from other one in terms of environmental condition, safety, and lot of things. So that's why we train a day before, like day 2. We just train. But for the T maze, the Y maze, the open feed, there's no training. It's just basically they just come. But what we do is that a night before, we place them in the environment where the neurobehavioral test will be conducted. A night before. So it's just to acclimatize them to that environment. That's just the only thing. But the training is just done for the Maurice water maze to be specific, and it's based on a protocol that we've seen that has worked over the years, and based on research evidences that it's better to train them a day before. And basically, you're not training them now and they are starting in the next 2 hours. No, you train them today, then tomorrow they start. So once they get into the water, they'll be like, oh, I was here yesterday. So they find a way to get their way out of there. Yeah, so that's how it works. And it does not really affect the results, especially for Maurice water maze, because they are all trained on equal grounds. Yes, and specifically at day 3, if you have any negative control, you might not really see much effect, yeah, at day 3. It's very, very rare when you see a lot of effect at that day 3. Most of the time, the effects are being visible at day 7 in terms of neurobehavior. As far as our own study was concerned, it was day 7, day 14 that we started seeing those effects. So at that early stage, you see little to no effect. And also, it does not really affect the results per se. Yeah, but acclimatization is very, very important thing to do during neurobehavioral tests. You place them in that same environment where the neurobehavioral test will be conducted the night before. Yeah, thank you.
Thank
So
you.
I hope that answers the question.
Yeah, that helped a lot. I actually didn't know I didn't realize that the rats are pre-trained. But I had another question. I remember you were saying that you only dissected 3 rats from each group for the biochemical analysis. And I think you were saying why, but could you explain again why you only dissected 3 of the rats and not all of them from each group?
Okay. So what happened was that we had 10 rats in each group, right? So for the neurobehavioral, during the period that we made use of 6 to 5 rats. 6 to 5 rats. So after the neurobehavioral, the truth is that for the potassium dichromate in particular, some of the rats died. Yeah. And some of them died at around day 21 to day 28, towards the end of the study. So at that end, we had just around 6 to 7 rats left. So what we did was, for the immunohistochemistry, we made use of, I think, 3 rats. For the biochemical, we made use of 4 rats. It was still the same sample. Then we did sonication after the sampling processes, neurotransmitter. But what we just did was we had a minimum of 3 to 4 rats for each of the laboratory studies that were conducted. That was what we ensured. But for the neurobehavioral, at least minimum of 5 rats.
Okay. Thank you.
Yeah. Thank you very much. Yeah, it's very much common. Rats die, and sometimes rat die even without administering anything. I can tell you for sure, we had some rats, like I said, we purchased 80 rats. We made use of just 70 rats. We had 10 as extra when we were just giving them normal food. Why we did that was we wanted to be sure if there was any effect in terms of environmental effect that could have affected the rats ordinarily. So, but we saw that even out of the 10, one of them still died. And why they died was you can just wake up in the morning and just see one is just dead. So in some scenarios, the environmental condition may not be quite okay for them. But in a scenario where you have 10 extras that you're not even making use of at all, you're not administering anything, and you just see them dying, then you have to question the environment, because the environment can pose a lot of risk to their well-being. So that was why we had the extra. So in scenarios like that, they can also develop conditions too. If their feeding troughs, where they drink water and everything are not cleaned properly. So you need to ensure that you give them clean water, and their food, the standard trough. Yeah, we need to, those are one of the things too. So, but when you see high morbidity, and that's why you need to conduct pilot study before you engage in your proper research. So you can have an understanding of what you are going to expect. And if there are any modifications that you can conduct early, you do them. So that was what we did on our own end. And we also had another group where we did not administer anything, even because the normal saline that we're administering, you might feel it's perfect, or the water. And some of them might die, although none of them died for our control group, but I'm just giving an example. So we had another control where we're just feeding them and just seeing the effect of the environment on them. So if we wake up in a day and we just see like 5 of them died in that group, then we know the environment is not suitable. Then we have to stop the research immediately. So that's basically one of the things that comes with it. But the rats are very good animals. For those of us that work with them, there's no way we work with them and at some point you want to dissect. I mean, when it's time for dissection, you have this connection and emotional feelings. I mean, these are people that you've been with for about 28 days, and now you have to dissect. But science has to prevail, so you just have to do what you need to do. But yeah, they are very, very good animals, and animals that I really enjoy working with. Yeah. So okay, if there are no questions, I believe okay. Yeah, thank you very much. Thank you very much. Okay, so but for me, if there are any things we would like to ask, you can direct it to the. I'm always open for questions, and also, like I said, what I'll be sharing are two different things. We don't really have enough time, and we really do not have that environment where we can do a proper video of how the immunochemistry is like. Because for me, I didn't appreciate immunochemistry when it was taught theoretically in school until when I actually did it and I've engaged a lot in it. Yeah, that's when I appreciated how beautiful it is, especially when you are viewing it on fluorescence microscope. You see the neurons and you know that neurobiology is very, very sweet. So I would have loved maybe in one of these times when we have when there's time, you can create a session for that. I myself I'll plan towards it and also inform my PI so that the necessary reagents that we'll be using for it will be made available. But if you have any questions in terms of image J, I will try to send the file on the Google Drive so that we can check it out. So if you have questions, we can go through it. But for the GraphPad Prism, you have to subscribe. So most times, if you are in a school or a lab, they should be able to sort that out for you, or a mentor that can sort that out for you. So that's how it works. So thank you very much. But before we go, I would like us to do something. We have to take a group picture. So I will appreciate if you can all turn in our videos. And I don't know if that's allowed. My hosts, is it allowed?
Yeah, it's allowed.
Yeah. Yeah, so we'll do that right now. Everyone, please, we can just turn in our video so that we can take a group picture.
Thank you
Wow,
for the
it's glad to see all of you.
presentation.
If we have time, please, can we all just introduce ourselves?
Thank you for the presentation. I think
Yeah,
sometimes
I
you
would
were really
appreciate that
funny.
all of you introduce yourselves. It's very nice meeting all of you.
Oh, I think we can't hear you. I think we can't hear you. I think we can't hear you.
Okay. Like I said, I've been following International Neuroscience Association Gharbiya chapter for quite a while, and you guys are doing amazingly well. I must commend you. I mean, I think you were the best in the last best chapter, if I'm really right. Yeah. So you guys are really doing well, and I must commend everyone, the executives, the members, and everyone. And I look forward to more collaborations. We ourselves, that's at IYNA. More affiliated with IYNA, but um, so far so good. I've joined International Neuroscience now, and I'm part of what we are doing, and we are trying to set up a region at Nigeria here and see how we can collaborate and do more, because YNAN so far has been the association that has been pulling out things here in Nigeria, but we are integrating it now and we are in course full collaboration now with International Neuroscience Association. So thank you everyone for joining. If you have any question, please reach out, or I can just put in my mail so that it's not going to be so hard to reach out to Tolulope was like issue for the that's so yeah. So that's my official mail. If you want to reach out, just put the subject International Neuroscience Association Gharbiya chapter as the so I'll send every other thing I need to send. The immunohistochemistry procedures, I'll send it, and the glutamate assay procedure, I'll send it to. So, and the PowerPoint itself, for I don't know, it might be useful in any other day for any one of us, so we might know use of it. Thank you very much once again. I really appreciate this privilege and opportunity to share and interact with you all. And thank you very much. See you all later.
Thank you.